So thanks, Sharif, for presenting today. I can say a very few words about you before we start. So Sharif is a postdoc in the group of Peter Panettini at NIH. He's a neuroscientist. He cares about consciousness and did his PhD in Yale, I believe. Sharif looks at consciousness with a very smart paradigm. He uses afterimages, and I think you will introduce them. I'm not sure if you will talk about it in your presentation, so I'm saying it that you're looking at this paradigm with many different modalities, like pupillometry, eye tracking, MEG, and I think you will mostly talk about the fMRI part today, I believe. Yes, then please share your screen and tell us about it.
Thank you. Okay, so hopefully everyone can see my screen. Fantastic. Okay, good morning, everyone. Thanks for joining. I really appreciate you being here. And thanks for the introduction, Renzo. Indeed, my name is Sharif. I'm a postdoc in the National Institutes of Mental Health in the section on functional imaging methods. And I'm really grateful for this opportunity to share with you this project that is now at a very important stage where we're sort of dotting the I's and crossing the T's on the manuscript. So your feedback and your reflections on what I'm going to share is going to be actually very valuable as we put sort of the final pieces of this study together and submit it hopefully for peer review no later than next month. So what I'll be talking about today is afterimages. And there are many ways by which I introduce this work. If I'm with an audience that is interested in consciousness like me, I'll talk about it one way. With you all, I'm going to just try to impress upon you that afterimages are interesting and worth studying. And there's a mystery that has been ongoing for literally hundreds of years that we're trying to help resolve in this experiment. So let me first remind you what afterimages are. I know you've probably experienced these dozens, if not hundreds of times in your life. These are common visual illusions where you continue to have the impression of vision even without there being a simultaneous light source that is corresponding with what you're experiencing. What typically happens is you have a preceding light event, like you're looking at a bright image, and then that image goes away and you have sort of the imprint of that light source, that preceding light source that continues in your vision. There are generally two categories of afterimages. There are negative afterimages where the afterimage is a negative of the inducing visual event. So negative in the sense that it might appear as a complementary color or might appear in a opposing shade. And then there are positive afterimages where it appears in the same shade or color as the inducing event, the afterimage does. So let me give you an example right now of a negative afterimage. So all I need you to do is to fixate on the plus sign at the center of the screen. And then what you're going to see momentarily is a face silhouette image appear. Stay fixated the whole time. It's going to go away. And if you're like me, you'll notice sort of a ghostly, gray, whitish afterimage appear in its place. So we'll do it one more time. So I would say most people experience afterimages almost all the time, but we discovered that some people are a little more resistant to forming afterimages, and maybe you're one of them if you're not noticing that. So the big question that has been presented about afterimages for, again, really hundreds of years is summarized in this question here, which is, are afterimages retinal or cortical? In other words, do afterimages originate by some retinal process, or are there cortical origins to afterimages? And I think the reason why this question has come up long before modern neuroscience and neuroimaging technology is that our experience of afterimages fits within this liminal state between our normal vision and those of hallucination and imagery. So there is the extent to which we understand afterimages, this link to real-world visual stimulation. Something happens, it goes away, and then you have an afterimage. And in that sense, it feels like vision. But then at the same time, the afterimage is happening when there isn't any visual stimulation in the world. You can have an afterimage in the total dark. You can have an afterimage with your eyes closed. And to that extent, afterimages feel a little bit like a hallucination or like mental imagery. And so I think that dichotomy has resulted in this discussion of what exactly is the category of an afterimage. Is it more like seeing, or is it more like imagining or hallucinating, right? And to sort of to stir the pot a little bit, to build the intrigue here, there's been centuries of behavioral research that create a narrative about afterimages that doesn't fit the canonical notion of normal vision. So here's two examples of studies that highlight that you can have afterimages from an illusory event. So in the above, this filling in illusion can induce a filled in afterimage. Likewise, you can have afterimages that spread into locations in visual space that there wasn't ever a previous light source. So again, this sort of builds the sense that afterimages aren't just an imprint on the retina or some sort of recapitulation of what the previous light source was, but can be modified from that. There are even, just this is for fun really, but there are some stranger studies that, for example, this one-off study that said you can condition afterimages. So by pairing a tone with a light source and then abruptly removing the light source, people can have this illusory experience of something like an afterimage. And then stranger still, there are several studies with a giant grain of salt needed here that suggests that you can have afterimages from very vivid mental imagery or even dreams. We contributed a little bit to this work where we showed that afterimages are correlated, the various afterimage perceptual details like the contrast of the afterimage or how bright it is, the sharpness and the duration is correlated with the person's self-reported visual imagery vividness. So all of this is sort of building a little bit of a story or an impression that afterimages are linked with some of these categories of experience that we consider sensory independent. And then for this audience, I'll share this important detail, which is that it's not surprising that afterimages being such a longstanding intrigue for so many years. In fact, a fun fact is Darwin's father, his PhD thesis was on afterimages, and many other major, parents and grandparents of modern neuroscience reflected on afterimages. So noting that historical relevance, it's not surprising that when we come to in the 80s and early 90s, the wonderful invention of MRI and fMRI technology, one of the first ever human fMRI studies was to study the neuromechanism of afterimage. And here it is, 1995, and it was a single slice study, EPI, recording with 4 subjects. And you can see there's pixels here suggesting bold responses in visual cortex and fusiform gyrus linked to afterimages. But then something strange happened, which I don't have an explanation for, which is that after this study, there are a grand total of 2 more afterimage fMRI studies that were conducted, and all only report signals in the visual cortex, visual occipital regions. So for whatever reason, there wasn't an attempt to really explore in a deeper way the whole brain mechanisms of afterimages. And even, of course, back then they wouldn't have the approaches of layer resolution fMRI to give us a sense of the information flow dynamics. So that brings us to the current study and what we're trying to do here. We had 2 main aims. The first was to map the whole brain dynamics associated with afterimages. And the second was to dive into the cortical layer dynamics in primary visual cortex that might give us information about the information processing that could enrich or corroborate things that we're seeing in the whole brain responses. So that's what I'll be sharing today. As Renzo mentioned, we have also other modalities that we explored, in particular, eye tracking and pupillometry. I'm not going to talk about those. I have those as supplementary slides just in case there's a question about those results, but those are actually enriching in the end for the fMRI responses as well. So before I dive into the first aim, let me just take a quick pause and make sure there aren't any questions at this stage. And also, I just want to encourage everyone, if you do have a question, just interrupt me. I tend to do a bad job of monitoring the chat, so speaking up would be helpful. Okay, so let's start with the first aim. So this was a whole brain study of about 35 subjects, 70 there's our TR and our voxel size spatial resolution, and we were recording concurrent eye tracking and pupillometry. Let me first tell you about the behavioral task that we had participants doing while they're in the scanner. Unfortunately, this is not a very straightforward task, so just bear with me and do ask questions if I'm not clear. You got to understand this so that we can understand the remaining results. So, of course, we wanted a paradigm in which subjects were experiencing afterimages. So the way we did that was just like what you experienced before. We had a inducer stimulus, which is that face silhouette you saw earlier. That silhouette would appear, it would disappear, and then nothing would appear on screen after that. What I'm representing in the dotted circle there with the afterimage label is just a representation that subjects would report often that they would see an afterimage positioned on screen exactly where the inducer was, but nothing is on screen, just to be very clear. Now, an important goal of our study was not just to understand the mechanism of afterimages, but we wanted a control or a comparative group or condition that would allow us to help interpret what we're seeing in the afterimage responses. So that motivated the creation of what I'm going to call a mock afterimage. A mock afterimage is a real image. It's physically present on screen. But long story short, we had people report on what their afterimages look like, and we used that information to construct an animated image for each individual person that resembled their afterimage. And also to create or mimic the pairing that exists between the inducer and the afterimage, we also introduced what we call a mock inducer, which was this flashing stimulus that was designed to suppress the creation of afterimages. But then what we could do is present subsequent to that mock inducer, the mock afterimage, which was that subject's image, animated image that resembled their afterimage. So the goal here was to have, as best as we could, two perceptually similar events in the instance of the afterimage and the mock afterimage, but the origins or the sources associated with those events being distinguished. The mock afterimage coming from a stimulus that's actually on screen simultaneously, and the afterimage being induced somehow by the preceding inducer event. So this is these two image types that we had. So I'm going to show you an example of a mock afterimage trial, but here's the layout of it. So we have the fixation point, we have the mock inducer appearing, and then we show the mock afterimage with this physically present stimulus, and then a rest interval. And then ultimately, what this lands on is we have three main event types or trial types. We have a trial type where there's just an inducer or a mock inducer and then nothing else. They perceive nothing after that. There's not an afterimage and there's not a mock afterimage. And then we have our mock inducer and mock afterimage trials as depicted here. We have the mock inducer and then the mock afterimage. And then we have our inducer and afterimage trials as depicted here, where you have the inducer followed by the afterimage. So those are our three trial types. And this yellow is just meant to highlight that our goal was for the mock afterimage and the afterimage to share perceptual features with each other, to look similar. And I'll just share quickly, in case I forget to mention it later, we did query our subjects about what they were experiencing, and the majority of subjects believed that the mock afterimage was illusory. They didn't think it was an on-screen event. They thought it was a hallucinatory like event, like their afterimage, as a point of evidence that those mock afterimages shared perceptual features with the afterimage. So let me show you an example of what this mock afterimage looked like. So what you're going to see, you don't need to fixate this time. What you're going to see is on the left side of the screen, the mock inducer appear first. And then subsequent to that, you're going to see this ghostly looking image, which it might resemble a little bit of what your afterimage looked like before. But remember, this is for just one subject, and every subject had their own mock afterimage. So there's the mock inducer, the flashing thing, and then now you see this mock afterimage stimulus appear. Okay. So let me take a pause there because I know this is a strange part of the paradigm. Do you have any questions about the paradigm before I show you the results we care about?
Maybe I have a question. Why faces?
Yeah, good question. So typically, when you go into the afterimage literature, you find that they often use like geometric shapes or like spots of light. And we're interested in using faces. One, there was like a curiosity that we had originally when we were doing our behavioral pilot, which is, can the afterimage maintain a detail of the inducing stimulus perceptual content, right? Does the afterimage always look like a blob, or can it maintain sort of a perceptual feature like the identity of the stimulus, like a face or something like this? So that was one curiosity. But really what we were hoping to do is drive as strong a signal as we could. And we know from previous work that the more complex the stimulus is, the better or the stronger a signal could be that we could generate from these events, because we knew the after images would be very subtle. So in the end, that we wanted to pick something that had some sort of content associated with it, like a face that has this very special representation in the brain.
We have a question in the chat. You said you're not checking the check. It's Lars Mukli asking could you show the mock after image again, please?
Yes, of course. So it starts with the mock inducer, and then once the flashing thing goes away, now you might notice just subtly the mock afterimage appears. And again, I'll emphasize that every person had their own unique mock afterimage. So some people with brighter afterimages had brighter mock afterimages. People with dimmer afterimages had dimmer mock afterimages.
Sharif, can I ask you one more question?
Please, go ahead.
Why you present this after image in peripheral or in your left visual side instead of the central or other part of your visual field?
Yeah, good question. So we presented, I didn't mention that we counterbalanced the presentation of the inducer and the afterimage and mock afterimage on the left and the right side of the screen. So it was both on the left and right. Why we didn't put it right at the center is because we were not sure how the fixation point would interact with the mock inducer, or I'm sorry, the mock afterimage. So it's a funny thing of, when the mock afterimage shows up, do you sort of place it on top of the fixation point, or do you blend it into the fixation point? We would need to match what people were experiencing with the afterimage, which again, I don't know the answer. When you see an afterimage on top of another image, do those two experiences sort of mix together, or is it sort of like a sticker that gets added on? So because of that complexity, we just wanted to keep the afterimage located in the area that was blank, where we didn't have to deal with some of this complexity, and likewise with the mock afterimage. So we put them on the sides of the screen.
Yeah, thank you.
Yeah.
Atana Akbari is asking about the order of these images, if they're random. I guess you're referring to different trial conditions, right?
Yeah, all the trials were randomized. Yeah, equal number of trials, but randomized.
Yeah. One more question, sorry. Could you please remind me of why you need this mock after images? Is it just to distinguish between the hallucination and the real effect of the images?
Yeah, that's basically the idea. We wanted to be able to sort of an implicit question that we're answering here is, are after images like images? Are after images like seeing something that's physically present, in the world? And so we wanted that control condition to compare our afterimage results to. But we didn't want to just show people any image, because then the differences that might show up could be related to differences in the perceptual content of the experience. Like if we just showed a house versus the afterimage of a face, or even if we showed a face, but the face was very bright relative or longer lasting relative to the afterimage experience. So we did our best to try to just match the experience of the afterimage in an animated image that could offer us, as close as we could, a control of what a real stimulus or a physical stimulus control to the afterimage. Yeah. Excellent. All right, thanks for all these questions.
Maybe one more.
Yes.
Thanks a lot for this. But I have one question. Like the hypothesis would be that it's a retinal effect, right? And so I was wondering if you could maybe create a retinal after image that does not come with a cortical perception. So, for example, doing, I don't know if that works at all, but do binocular rivalry, in which the suppressed stimulus creates something like a retinal after image. So we have a face in a house and make sure that the house is flickering, so the house is creating the percept, but maybe the suppressed other stimulus creates an after image, and then you would know that that's from a retinal but not perceptual. Anyway, so just trying to think about how to control, because your control seems to be, as you just pointed out, very much aligning to the idea that you need to perceptually balance what you perceive. If it's stronger or weaker, you might get a difference, but that's really very difficult to do, right?
Yes. Yeah. It all is very difficult. Right. I don't think what we came upon is bulletproof to some of the critiques you're bringing up. In the end, if you don't mind, I'm going to table your question to the end, because I think we do want to share a perspective on this retinal versus cortical dynamic, which I think is more holistic ultimately. So let me continue and get to that at the end, if that's okay with you.
Sure.
Yeah. Excellent. Okay, so let me start with some quick behavioral results just to make sure that the paradigm is working as we hope. So what you're seeing in the red bar here is when we present the inducer and then nothing else, subjects are reporting on average about 75% of the time that they're seeing an afterimage. Subsequently, with, as I mentioned, some subjects, those circles at the bottom there being more resistant to this. When we show the mock inducer with a mock afterimage, and we ask them, hey, did you see an afterimage? Remember, most subjects think those mock afterimages are in fact illusory. Most subjects are saying they see those see an afterimage. And then when we just show the mock inducer with nothing else, so that flashing event, then you find that most people are saying, I'm not seeing an afterimage, just to evidence the point that our mock inducer isn't driving an afterimage, which is important because we don't want there to be sort of a mixing of events here. While the subject's in the scanner, they are telling us the onset and the offset of their afterimages and the mock afterimages. And what we can do as a way to give us some insight to whether the experience of the afterimage and the mock afterimage were similar is just comparing these temporal characteristics of these reports. So you can see here that the average duration of the reported afterimage is not different from the average duration of the reported mock afterimages, and the same is true for the onset latencies. And then if we do a correlation of the reported duration of the afterimage by the reported duration of the mock afterimage, you see a correlation across subjects, meaning subjects have similar responses for the temporal features of their afterimage and the mock afterimage. Another thing that we notice in the behavioral responses is that if you assess the within subject variance on the reported duration and onset latency, there is greater variance, within subject variance, for the afterimage versus a mock afterimage, meaning that each afterimage, even within subject, is a little bit different from other afterimages, more different than the other afterimage reported by that subject than for the mock afterimage. And this is a result I'll come back to at the end, but just highlighting that there is more, the afterimage experience seems to be more variable relative to the mock afterimage, which is this physical stimulus. Okay, so let me get to the results that we really care about. So these are whole brain statistical maps showing you t-values. Everything here is statistically significant as outlined by the black border. And we have 3 contrasts. What we've done here is we subtracted away the inducer and mock inducer effects. So what you're seeing is just those associated with the afterimage and the mock afterimage, and then the far right column is a subtraction. So let's just start with the far left. And what I'm highlighting is just a single time point, which based off of our observation of the data represents the most salient or the strongest, the peak time for the afterimage and mock afterimage signal. And you're seeing just, of course, axial and the sagittal view. And then we also projected these responses to a cortical surface to give you an idea of where these activations are. And long story short is you're seeing whole brain responses associated with these afterimages. Of course, there are elements of the signal that are likely, no doubt, in fact, related to the task itself, them reporting on the onset and offset, as you can see here with the left motor cortex lighting up associated with their right-handed finger presses for the onset and the offset. But areas that we might expect are related to the afterimage itself include regions like fusiform gyrus, lateral occipital cortex, primary visual cortex. We also get salience regions activated, including the insula, the anterior cingulate cortex, and then you see some deactivations in areas that we typically associate with the default mode network, like the posterior, the postcingulate cortex, and the ventromedial prefrontal cortex, which these responses could be associated with sort of the task engagement. When we look to the mock afterimage condition, you find lots of the same areas lighting up. So, some of the activations you might notice, we're going to highlight this in some other slides in just a moment, but you might notice the activations are the area is a little bit bigger, the signal size is a little bit bigger, but lots of the same areas are highlighted in the mock afterimage are also present in the afterimage condition. And then things would get interesting when we do subtraction between the two. So this is basically subtracting the leftmost and the middle column to get this right comparison. And what you're seeing here, there are basically these global decreases, meaning that the afterimage condition, while it is activating many of the same areas that you see for the mock afterimage, the magnitude of that response is weaker for the afterimage, with the exception of a few particular regions, including some salience areas that, as you can see here, the anterior cingulate cortex, and also some of the insula, as you can see there, which is greater for the afterimage relative to the mock afterimage. So let's dig into these maps a little bit more to help us interrogate what's going on. So the first thing that we did was we took all of the areas that were activated by the afterimage and the mock afterimage conditions, and we created a big ROI from all of those areas. It didn't matter whether it was an increase or a decreasing response. And we then applied clustering, k-means clustering in this case, to help us interrogate the different spatial temporal dynamics of these co-activated regions. And what we ended up with, we tested a number of different K values or different number of clusters, and using silhouette scores to help us assess the integrity of these clusters. We landed on 3 being the best clustering value. And we just defined these clusters according to generally their functional, the association of the regions within each cluster is to known functional roles. So in pink, we call this the sensory and salience network. In green, we call this the task positive network. And in orange or yellow, this is our task negative network, or you might otherwise call it the default mode network. So you'll notice that in the sensory and salience network, all of the visual primary and extrastriate and in higher order visual areas are clustered together. So you get primary visual cortex, fusiform gyrus, LGN even lands in there. Here you can see primary visual cortex being punched out. And then the salience areas include the anterior cingulate cortex. You see the insula being pulled out. And then you also have some areas that don't necessarily fit perfectly in our definition, like the left motor cortex and some posterior parietal attention areas. And then with the green, we call this TAS positive, and you get basal ganglia, nucleus accumbens, prefrontal cortex, and then the TAS negative network or default mode, you get areas like the precuneus, posterior cingulate cortex, angular gyrus, the medial prefrontal cortex. Helping us to understand these clusters are these time courses you're seeing below. So this is time and percent change bold along the y-axis. And what we basically did was we just took each of these clusters and we just averaged the bold response across time for all voxels within those clusters. And you see very distinct response profiles. So let me start with the most obvious. We have the default mode, which is the orange or yellow, which shows you this decreasing response. Oh, by the way, let me just say that the bars here represent the proximate timing of our events. The first gray bar is telling you the timing of the inducer and the mock inducer. And then this second gray bar with the dotted outline is approximate timing of the mock afterimage and the afterimage. So the TAS negative network shows you this decrease response here. And then there's almost a mirrored image response associated with the sensory and salience network. And then you get this late peaking response associated with the TAS positive network. And that makes a lot of sense because the subjects are making a behavioral response to the events associated with the second gray bar, not the first. So the response in the green is associated with those later events that are occurring. We can then take these ROIs or these clusters and treat them as ROIs. And then remember those three trial types we had. We can apply the response associated with those three trial types within these networks. And so this is what you're seeing here. So we have the salience sensory network, the TAS positive network, and the TAS negative network. And then these three lines, the light blue line is showing you those trials where there's just an inducer or a mock inducer and nothing else. There's just one event, right? An early event. The red is when there's an inducer and the subjects report seeing an afterimage. And the dark blue is when there is a mock inducer and then there is a mock afterimage present. So basically, light blue is one event, red and dark blue are two events. And that really is present in the response profile, as you can see here in the salience and sensory network, where you get this more transient response with the light blue curve, while there is a prolonged response with the red and the dark blue responses or curves. This is percent change bold again. When we go to the TAS positive network, you'll find, as expected, because people are responding to afterimages and mock afterimages with button presses, you get this late response for the red and the dark blue, but not for the light blue because they're not responding. They're not making a button press for those trials because they're only asked to respond when they either see an afterimage or mock afterimage. And then you see similar responses going on in the TAS negative network. So what this told us is that the network that we really care about is the salience and sensory network, because the TAS positive network was more about responding to the task, and the TAS negative network was negatively responsive or decreasing for the events we're interested in. So the areas that were responsive to the events that we care about are in the salience and sensory network. So we took that network and then we broke it down into representative ROIs. And that's what you're seeing here. So all of these regions were in that salient sensory network. And I'm highlighting several areas of interest here, which we'll go through in detail. So we have areas within the primary visual pathway from LGN to fusiform gyrus. That's the second column. And then we have some salience areas like the anterior cingulate cortex and the insula. And then as a control, I'm also presenting the left motor cortex And let's just start there. So let me just be very clear on what I'm presenting to you. We have time along the x-axis. We have percent change bold along the y-axis. Remember that first gray bar is the presentation of the inducer and the mock inducer. That second gray bar between 6 and 10 is the approximate period when the afterimage appears or the mock afterimage appears. And we have those three trial conditions again, same colors as I shared with you earlier, light blue, dark blue, and red. So not surprisingly, in the left motor cortex, only the light blue and the dark blue traces show a response. Those are the conditions where the subject is making a button press response. They're not making a button press response in the light blue condition, and you don't see a response there. And also importantly, you'll notice that the red and the red and the dark blue response have similar amplitude signal. Right? The response is similar in size. And that is important because we find a different magnitude response between these conditions in other areas. So let's start with the LGN. So here you'll see there's like this bimodal response, no doubt related to the inducer event associated with the first response, and then the mock inducer and afterimage event, which is the second response. And it's not as obvious, but it becomes clear going into the other visual regions where you find that the mock afterimage is consistently, that's the blue trace, has a consistently stronger amplitude response than the afterimage. So the dark blue is bigger in the second peak than the red. And that's what we were seeing in the whole brain maps as well. That's particularly obvious when you go to primary visual cortex. So you get, importantly, that first response associated with the inducer, mock inducer are very similar. Only when you get to the period of time that's responsive to the mock afterimage and afterimage do you see this separation where the dark blue is stronger than the red. The mock afterimage has a bigger signal than the afterimage. And you also see that same pattern of response in fusiform gyrus, although now that double peak has sort of been squeezed into this, unimodal response, but is more prolonged relative to the light blue trace. Okay. Now, remember when we looked at the brain maps, we also noticed that there were some areas that were reversed in the amplitude response, meaning that the afterimage was stronger. And I'm highlighting those areas here, the anterior cingulate cortex and insula, where you can appreciate where we have this red afterimage response being larger than what we find for the mock afterimage. That's the blue. So let me take a quick pause before I get to the layer results. Okay, so let's move on to some layer results. So I'll quickly summarize what we think we're seeing in the whole brain. What we think we see is that by and large, afterimages involve some of the same areas of the brain that images do. When you see an image and when you see an afterimage, the same areas are activated, but in most cases, the afterimage signal is weaker. At least the bold signal, we can say, is weaker than the image-related signal, with the exception of some areas of the salience network. So I'll provide some interpretation of what we think that is, but that's sort of what we are seeing, or what we wish to summarize from these findings. So the next goal, as I mentioned, as I began the talk, was to understand a little bit more, well, if we see similar areas of activation, indeed we saw primary visual cortex activated in both afterimages and image conditions, does that necessarily mean that the way that the information is being processed is the same? And this gets to a little bit of this question of retinal versus cortical. Is the signal coming from the retina just as for images, but it's sort of a latent or perseveration of a retinal response in the case of afterimages. So we took some of the same subjects that we had complete the whole brain study, come back and do this V1 experiment with a higher resolution that would give us layer resolution information. Because this is a layer meeting group, this is a supplementary figure in our manuscript, but I won't go through all the details here, but just to share that Renzo and Barak Aiken put in a lot of work over years of trying to put together a processing pipeline that was optimized for this paradigm. But as all of you are likely familiar, long story short, is we extract the gray matter rim, we cut it into layers and columns, we arrived at activation maps associated with the events of the study and identified a primary visual cortex ROI, and then emphasized or studied the layer responses for bold and vaso within that ROI. I'm going to highlight the vaso results today, but we have the bold results. So step one is let's just make sure that our V1 layer resolution sequence can recapitulate the results that we were just seeing in the whole brain, because we're using the same subjects and the same paradigm. So we should see similar results. So this is a representative subject. You can see here is our slab that we acquired and the axial view from this dotted line along that slab, showing you the primary visual cortex ROI and the layers. And what you're seeing here is for that same subject, here's time, here's the percent change bold, the exact same type of maps we were seeing before. And very nicely, this subject, and again, this is representative of what we saw with other subjects, shows this double peak profile, just as we saw in the whole brain, where the first peak is associated with the inducer and the mock inducer, and the second peak is only present for the red and the dark blue. That's the afterimage and the mock afterimage, and not for the light blue where there's just that single event at the beginning. So that's a nice sanity check and link to the whole brain results. So let's look at some of the layer results now. So what you're seeing is percent change vaso along the y-axis, and time, the time points are going to be shown at the bottom of this figure once it all shows up on screen. So that's along the x-axis. Each of the traces represents the response associated with a layer, and you can see the color bar here representing layers, light blue, dark blue to red, red being closer to the CSF boundary. What you basically, I'll first highlight that all the layers are responding to at least the inducer events. So you can see all the layers are increasing. But you do see that the superficial layers are responding with a greater magnitude than the deeper layers. And then if you look after this dotted line period, this is where things start to change between our after image, that's C, versus our mock after image condition, where you'll notice that the superficial layers continue to dominate through the response, while for the after image condition, the deep layers begin to dominate. And when we do a subtraction, this is really nicely highlighted here. So at the time period, exactly when we would expect for the afterimage and mock afterimage signal to be most prevalent, you see that the afterimage signal has a stronger deep response, deep layer response relative to the mock afterimage. And another way to look at it is a more holistic representation of these findings. Just start with the middle, this top row of maps. So this is again time, but now along the y-axis is layer, also organized from white matter to CSF. And the color is showing you T values associated with a spatial temporal test that we performed on the percent change vaso responses. And then the black outline is showing you statistically significant spatial temporal clusters. So in this case, the spatial temporal cluster is time by layer. So again, what you see is that there, for both the afterimage and mock afterimage, you get a very broad response across all layers. That's that big yellow band that persists between 0-ish and 6 seconds. But then when you get to the period of time that's responsive to the afterimage, you can see that it's the deep layers that are significant for the afterimage, while it's the middle and superficial layers that are significant for the mock afterimage. And then when you do the subtraction, that's H, you see that at the time period around the afterimage and mock afterimage response period, you get a significant increase in the deep areas relative to the mock afterimage. The bottom row is showing you the exact same data, essentially, as the top row heat maps. The difference is now we've normalized the layer response across layers, which gives you just a different perspective or a different look at the response. So I'll start with highlighting actually the mock afterimage response in this instance. So that's J. And what you see is there's two almost mirrored image signals that we're getting. There's this one that sort of has a peak around 5, and then there's a little bit of like a break, and then there's a secondary response that peaks around 12, right? But when you look at the afterimage, you don't get that mirrored image response. And I think what that's telling us is that for the mock afterimage condition, you have the mock inducer, which is a real stimulus, which generates a real or on-screen stimulus like layer response. And then the mock afterimage is also a real stimulus. So you get a similar looking response. But for the afterimage itself is different. It has a different layer profile, different layer pattern. So you only get that first response that mimics something like a on-screen stimulus, and then it's a different response pattern later. Okay. So let me just, I'm sure there's questions, and I could explain that more clearly. So let me just finish, and then we can just have time for questions about any part of it. So how are we trying to like piece all of this together? We don't want to be sort of grandiose here and say we solved the mystery of afterimages, but I think these results do offer a framework or a model to help us integrate not only the results that we saw in this study, but also some of the previous behavioral results I was presenting that suggest that afterimages are susceptible to top-down and bottom-up influences and state-based influences as well that have been used in the past as evidence that afterimages are cortical in origin. And here's the basic idea of this framework. When you are presented an image, particularly a very strong image, you have a strong retinal input associated with that image, and that drives a feed-forward dominant response. And that means that there are, of course, ongoing top-down and bottom-up influences on that signal, but that strong feed-forward response is just more resistant to effects like state-based effects, priming effects, prior effects. And we know this from the image perception literature. As the image becomes more degraded or your perception of the experience becomes more liminal, it becomes more susceptible to top-down and bottom-up effects. And what this means is when we get to the stage at which, that magical black box of where perception comes out, the experience is consistent within subjects and consistent with the stimulus that you experience, right? So when we look within subjects and we look across subjects, the experience associated with that image is consistent. And that's what we saw with those behavioral results before. The mock afterimage reports were consistent within subjects and across subjects. But what's going on with afterimages, perhaps, is as we saw in the bold response, the signal is weaker, inherently weaker across the primary visual pathway. So that might mean that we don't know what's happening in the retina. We didn't record the retina, right? But based off of what we saw in the LGN and V1 and in FG and other visual regions, the suggestion here is the signal might be weaker from the retina from the beginning. But in any case, it's weaker once it's in the brain. And that weaker signal means that it's just more susceptible to state-based effects, prior effects, priming effects, contextual effects, all these previous behavioral results that suggest this. And that might explain why there's just more variability across subjects and within subjects on the perception of their afterimages, because every single event is going to enter a new state of your physiology or have been primed or priored in a different way for every particular instant of that afterimage event. So This is one way that we think we can try to put this all together, and this is in part corroborated by the layer results. But take it or leave it. You don't need to abide by this framework. The results I just shared could exist within other models or frameworks. So let's just wrap up with this final question, which I began with here, which is we've all been asking for a while, or at least historically, are afterimages retinal or cortical? And we want to say that this is a false dichotomy. In the end, this is not an appropriate way to frame the afterimage study. We don't ever talk about image perception this way. No one says images are retinal, right? We understand that, yes, there's a retinal input, but there are cortical contributions that affect the signal as well. And we think afterimages are probably no different. So we can put the retinal versus cortical debate to bed and just understand this as we would any other perception, like image perceptions. Okay, I'll close there. Everyone in blue are the very important collaborators on this project. It took several years and now reaching, hopefully, a final conclusion here. And then, of course, thanks to all of you for your attention, and happy to take any questions.
Thanks a lot, Sharif. So we have time for questions, a bit time. I saw a couple of people switched on the camera. Jeff Dunn has a hands up. Jeff, do you want to start?
Yeah, can you hear me?
Yes.
Yeah.
Oh, a really nice presentation. I really enjoyed it, Sharif.
Thank you.
A few questions. So one is one of my favorite topic, arousal, or maybe what you mentioned, task engagement. So when people have to press the button when they see an afterimage, they probably get aroused or excited. That affects neural activity, but also autonomic activity. My question is, have you corrected for that? But I'll keep going with my questions because they're kind of related.
Sure, go ahead.
So the other one is, I noticed in so you presented task in the periphery, and there was a comment about that. Why not in the fovea? And I sort of feel like the periphery is kind of a different beast compared to normal visual processing, perceptual visual processing. But what you showed in your results is that both sides of peripheral V1 were activated, and your stimulus is only on one side. So have you compared left versus right side, which may also get at the point of this arousal effect, because the arousal effect is mostly uniform, bilateral. And then the third question, also a little bit related to that, is why did you use this task design? I don't know. You probably don't want to hear this question. Why didn't you use a noise scrambler after your inducer? So with noise scramblers, they can be very brief. You can eliminate the afterimage effect, and it can be a bilateral noise scrambler. So you will have a still. You can do a noise scrambler versus a non-noise scrambler. In the non-noise scrambler, you get an unilateral afterimage, and in the noise scrambler, you get nothing. So you can, or you get some bilateral effect from the noise scrambling. But you could correct for the noise scrambler by looking at the other side in V1. So why didn't you use the noise scrambling? But so my main question is left versus right. Why didn't you show any of that?
Okay, all great questions. So let me think about how to start. So let me start with the arousal question, actually, and I have some slides to share that facilitate an answer. So you asked basically, how are we controlling the arousal response? We agree that there would be arousal response. Our approach in this regard was, because we're contrasting in the end the afterimage from the mock afterimage, they are responding to both. So
yeah, but the afterimage is more exciting, right?
Well, yes, but the idea was for the mock afterimage to look like the afterimage. So it doesn't appear to be any different or more exciting in that regard. It could still be the case. And in fact, to your point, we are seeing heightened arousal signals, or I mean, I call them salience regions, associated with the afterimage. So that could be what you're saying. It's more exciting.
Right. But you could check it from the peripheral signals, right? From the cardiac heart rate or respiratory effort. You can check if that is stronger with afterimage versus no afterimage.
Yes. So let me share my screen again and then get to some of your other questions. So this might be a little bit helpful. Oops. This might be a little bit helpful to the arousal question. So this is showing you the pupillary responses. This is the top row. By the way, can you see my mouse at all?
Yeah.
Oh, okay. So here's the pupil response. And what's interesting is that you'll find that if we break up trials between trials where an afterimage was perceived, that's the red, and trials where an afterimage wasn't perceived, that's the orange. You see there's a significant difference there. The pupil is greater preceding trials where an afterimage is perceived. So, pupil size has been used as a measure of arousal and vigilance.
Yeah, sure.
That the preceding state can be predictive or be at least as correlated in that regard. So then let me get to your next question. So we did look at left versus right visual field presentations. And what you see here, so top row is left, bottom row is right. The first column is the afterimage, the second column is the mock afterimage. So in the visual areas, you find that you get the anticipated contralateral activation. So for the left visual field, you get right. Here you're seeing fusiform gyrus, extrastriate cortex, and primary visual cortex. And then for right visual field, it's opposite, right? The opposite areas. But then to your point, the anterior cingulate cortex has, at least that area is activated contralaterally, or bilaterally rather, irrespective of which location the stimulus is presented.
Yeah, okay, but that's understandable, right? It gets mixed beyond V1, V2, it gets mixed.
So I think there's an additional analysis that you sort of suggested that we need to do in this regard. But at the very least, what this tells us is that afterimages are following the rule of the primary visual pathway of, following those tracks. And we were curious if there might be sort of a spreading of the signal. Yeah. And we are not seeing that. We're not seeing that. And then finally, your last question, it was your approach of using basically a mask could be a totally legitimate approach. At the end, I'll say it was my concern. Using approaches like that means that the trials where there's an afterimage and trials where there's not an afterimage are different. There's a different stimulus.
Yes.
And we wanted to just try to match those two trials as best as possible. And so that's where we introduce the mock inducer and the mock afterimage, which has its limits as well. But at least we have identical, event times, and identical number of events. There's an inducer that lasts 4 seconds in both cases, and then there's an afterimage or a mock afterimage that have similar perceptual features and last about the same time. So that was ultimately a decision there, but at this point we could have thought about something different as well.
Okay, great. Thank you.
Thank you.
Are there any other questions? Yes, Lars has his hand up. Please, Lars.
Yes, thanks a lot for this presentation. I'm trying to get my head around how it relates to some of our data from Johanna Bergman, where we show illusory contours to be in the superficial layers and imagery of colors in the deep layers. And our interpretation is that it's more of a contextual, automatic feedback that reaches the superficial. And I think that's consistent with what you have been saying, that you have a weak stimulus and you're amplifying that. That's the typical visual system at work. And then in the other scenario where you have this after image, following our interpretation, it could be something like, so here so for us, it's imagery. The instruction is imagine a green disk, imagine a red disk.
Yeah.
And we could decode the color in deep layers. So the question is the after image something memory related? Do we keep it longer? Is there maybe other interpretation from dual task or something to say after image is a little bit like keeping something longer in your visual memory, and therefore might be using the same kind of microcircuitry that involves the deep layers? That was just...
Yeah, that. I really appreciate that comment, because you're now reminding me that we and two other groups have found a relationship between after image vividness and imagery vividness within subjects. So that's just a behavioral result that might help link to what you're speculating as being shared neuromechanisms associated with after images and imagery, and potentially distinguishing it from what you're saying as like filling in or hallucinatory types of things.
Very interesting. Yeah, that's an interesting. Yeah.
Thanks so much for bringing that up to make that connection.
Thanks a lot. It's already 5 minutes past 11 here. So if there are no further burning questions, I thank you a lot, Sharif, and everybody for the great discussion. And this will be on YouTube, and I sent the link around. So in 2 weeks, we will meet again with another presentation from Amalia, and this will be more sequence focused on kind of cortical movements and cortical thickness changes and velocity changes across layers. So see you in 2 weeks, everybody. Bye-bye.
Bye-bye. Thank you.